Hello everyone, and welcome to this audio podcast. This audio podcast is part of a Bulletin beat. This audio podcast is part of a Squadron Bloca, and this audio podcast as part of an an audio podcast as part of a PLCE Labonics. This audio podcast as part of Storm Robotics production. In this audio podcast, will be an overview in regards to the importance of reduced immunosuppression in pediatric renal recipients. In pediatric renal recipients, to prompt a sort of to prompt better prognoses in around allografts where renal or pathophysiology is concerned. So this audio podcast will look to cover the following. We'll look to cover the following. There'll be an intro to this works, and a little bit of an info in around transplantation, transplant immunobiology, looking at sort of an overview in around the MHC or major histocompatibility complex and human leukocyte antigens, HLA, and looking at immunological tolerance, looking at immunosuppression as a sort of main category in around a sort of a in around sort of the basis of the sort of therapeutics, the sort of therapeutics around immunosuppression, and then looking at sort of immunosuppression, the varying sort of immunosuppression sort of categories, and then looking at interleukin 2 receptor antibodies and monoclonal antibodies, looking at calcineurin inhibitors, and looking at sort of an overview in around immunosuppression sort of pharmacology, looking at biomarkers as part of this works, and a critique of laboratory medicine, and also there'll be as part of this works this sort of interlude. There will be a look at sort of a comprehending drug induced nephrotoxicity, and also looking at getting toxicology right, and then there will be an overall sort of summary. So to begin, in around transplantation. So retrospectively, by the end of 1995, over 250,000 patients or recipients with CKD stage 5, or where CKD stage 5 is and was or has been even sort of linked to ESRD or end stage renal disease, were alive in the United States. The number of new patients or recipients undergoing ESRD modalities have continued to increase at a rate of 7% to about 10% per annum or per year. And although not all end-stage renal disease ESRD patients or recipients are eligible for transplantation, in around the sort of mid 90s, nearly 37,000 patients or recipients were registered for possible transplantation, where renal transplantation or kidney transplantation was concerned, and over 54,000 people had been on the sort of total waiting list for all organs, However, in 1996, only 11,000 or just over 11,000 renal transplantations were performed, wherein specifically 3,389 transplants were from living donors. And for all of the various solid organ transplants, a disparity remained between the number of recipients waiting for transplantation, waiting for transplants and transplantation, and the availability of organs. And in around sort of 97, in around 1997, there were 3,900 or just under 4,000 recipients waiting for cardiac transplants. 2,700 for lung, for lung transplants, 235 for sort of double sort of transplants where cardiac and respiratory are concerned, cardiac and lung transplants, 9,600 for liver transplant, and around 92 for intestinal transplants. And in 1996, 2,340 cardiac, 805 lung, 39 heart or cardiac and lung, and 4,000 liver transplants were carried. And so, owing to sort of an increased experience and advances in sort of surgical procedures, tissue preservation, which have been one of the most important aspects, one of the most important sort of advances since those healthcare professionals had advanced in the way of ensuring tissues in around organs were sort of preserved or are preserved sort of applicably and appropriately according to varying organs that recipients had been sort of listed for and waiting for. So the advances in around the practices around tissue preservation had advanced, and post-transplant care, including IST or immunosuppression, the overall success rate of transplantation had been improving. So retrospectively at the same time in 1988, the one-year allograft prognosis using cadaveric grafts or allografts was around 76%, but by 1995, the one-year cadaveric graft or allograft survival prognosis increased from 76% to around 87%. And so results of renal transplantation from living donors improved from 89% in 1988 to 1993 to 93% in 1995. And so between live donors and cadaveric donors or cadaveric donors, the in between sort of the 1980s, late sort of 1980s to the mid 1990s, the prognoses, the allograft prognoses between live donors and cadaveric were actually sort of almost like for like. So with the increases in early allograft survival prognoses, more recipients experience better outcomes. So now leading into a little bit around transplant immunobiology. So unlike other solid organ transplants, the renal pathophysiology consists of ABO implied implied sugar antigens articulated on endothelial cells of blood vessels. Thus, if the donor recipient has a different blood group complex from a recipient, the antibodies will result in a type 2 hypersensitivity reaction in the renal allograft, thus causing an elimination of the transplant through cytotoxic T cells, i.e., for example, CD8 T cells, CD8 or CD8 cytotoxic T cells and as part of the sort of interplay with the major histocompatibility complex or the MHC, and T helper cells CD4 in around with the interplay with the MHC sort of are prompted sort of initially at least to aid in the recognition of transplants or the allograft, thus prompting a binding to explicit areas of the MHC. Grafted recipients or allografted recipients might also have some human leukocyte antigen or LHA through a rejection of a preceding allograft, and this applies specifically and probably perhaps more applicably to pediatric recipients that have had more than one allograft or more than one transplant. And so, as with other diseases that have a significant immunopathogenic element, a variety of renal illnesses have associations with the MHC. The first of these were to be have been sort of illustrated in works where there has been an association with an association amongst the DR2 antiglomerular DR2 being sort of DR2 sort of loci as part of the HLA and sort of the as part of the HLA and MHC, and in line to antiglomerular basement membrane disease. Subsequent several MHC alleles have been coupled with a variety of other renal diseases or nephrology sort of diseases. So immunobiology for MHC typing have advanced, but at the same time, whilst advancing rapidly from serological to sort of more molecular-based methods or molecular-based sort of modalities. And in complement to the MHC, several genetic indicators that may be linked with a variety of renal diseases had been investigated, but also solely based by solely from a research intrigue as far as evidence and published works has has uh, has uh, has documented. So now in around a little bit around the MHC and HLA. So the MHC is used as a basis of all solid organ transplants, and is thus part of underpinning immunobiology, and laboratory and sort of laboratory investigations. Recognition of antigen by T cell receptor or TCR requires MHC on the surface of antigen presenting cells. The APCs or the antigen presenting cells internalize large antigens, degrade them into small peptide fragments, and then pair up the fragments with MHC molecules, and then export the antigenic peptide or MHC complexes to surfaces where molecules interact with T cells. T cells recognize then sort of the antigen only when it is associated with an MHC molecule. T cells cannot recognize antigen free in solution like B cells can. This is and this is in literature. In literature, it's referred to as the LHA, the human leukocyte antigen region of chromosome 6. Class 1 antigens are present on the surface of most nucleated cells, whilst class 2 antigens are less broadly expressed, and the DI loci is equivalent to the D locus on the HLA. So this then sort of follows into immunological tolerance. So immunological tolerance. So scientists have been investigating transplant tolerance for more than sort of 5 decades, or over 4 decades, sort of where literature or evidence has indicated, to improve the sort of acceptance of tissue and cross-matched solid or sorry, the acceptance of a tissue and cross-matched solid for cross-matched for solid organ cross-matching for solid organ transplants. Self-tolerance, though, at the same time, can be partly explained by the destruction of self-tissue by T cells that react to self-antigens. However, some self-reactive T cells slip through the destruction process, destroying the host pathophysiology. And so, convinced by an antigenic specific mechanism of tolerance exists, early works from one team in around 2004, sort of retrospectively from the University of Washington, University of Washington and Rome began looking for and identified a different set of T cells. These T cells, or these suppressor T cells, induced tolerance after recognizing the antigens on surface of immune cells, and these were called dendritic cells, which process and present antigens. So somehow, after this sort of recognition, the T suppressor cell sort of robs the dendritic cell's ability to activate T helper and T killer cells to respond to the same antigen. And so without active T helper and T killer cells, the immune response to antigen is then put on hold. So looking at in early works again from the team in 2004, looking at the outcome of 15 cardiac transplanted recipients, the team had identified and found two sort of proteins that play a sort of pivotal role in vivo. The team informed the team had in sort of retrospectively again in around 2004, informed that 5 out of 6 recipients who had T suppressor cells were capable of suppressing the donor's dendritic cells, and wherein there was no allograft acute allograft rejection episodes. 9 other recipients could not suppress the dendritic cells, and all 9 experienced two to three acute allograft rejection episodes. So the team from 2004 retrospectively had ultimately identified that if dendritic cells could be suppressed, recipients may have a lower probable risk of acute allograft rejection. So now going into sort of looking into immunosuppression, or and throughout this sort of interlude, immunosuppression is abbreviated as IST. So transplant pharmacology advanced pharmacology has advanced where new immune IST agents are investigated, have been investigated and approved for use. Clinical immunosuppression, or IST again, has been sort of plagued retrospectively with maintaining a balance between rejection of a transplanted organ and complications over IST, including sort of leading infections and malignancy. And so whilst all transplanted recipients whilst all transplanted recipients may be on a form of IST, the combinations and choices sort of increase as new pharmacotherapies are trialed and developed. So the foundation for maintaining therapy rests sort of on combining IST to preclude rejection through a variety of pathways. And so an understanding of the mechanism of action and additive effects of pharmacotherapy allows professionals to optimize therapy whilst decreasing adverse effects. IST offers potential for reducing detrimental patient outcomes and improving allograft prognoses. Challenges facing investigators and clinicians focus on improved outcomes and constraints surrounding transplantation. So continuing on, during the past sort of decades, several new potent IST agents with different modes of action and different side effect profiles have become available. Nowadays, however, at the same time, IST after renal transplantation, for example, is no longer sort of a one single regimen applicable to all recipients. In the selection of optimal IST protocol, individual drug-related toxicity, recipient-related risk factors, as well as donor organ characteristics have to be taken into account, or have been sort of taken into account in context of the literature. A narrow therapeutic index unique to each recipient, as well as variable absorption, distribution, and elimination are characteristics of ISTs. So TDM, or therapeutic drug monitoring, plays a pivotal role in maintaining blood and plasma levels of IST within respective laboratory reference ranges. Variation in concentrations outside the narrow therapeutic sort of outside the narrow therapeutic the therapeutic laboratory reference ranges can result in adverse clinical outcomes. And so that's therapeutic monitoring, or therapeutic drug monitoring, TDM, of IST or ISTs has been based on several choices of assay and biological fluids. So, for example, in context of laboratory, and we go into, and there will be a slide in this as part of this interlude that looks at that we're looking at sort of the laboratory side of the laboratory aspects. However, IST TDM of ISTs has been based on several choices of assay and biological fluids. So, for example, for example, IST concentrations or trough levels, for example, can be determined best through whole blood or through plasma, for example, and appropriate for particular drugs. Particular in around particular ISTs, and TDM of one particular drug, or one particular IST, for example, FK506, which is referred to, which is another name, another sort of the sort of the pharmacological name for tacrolimus or around tacrolimus, for example, has been improved. Has been improved based on the development of assays and that have greater sensitivity and specificity. So now looking at sort of the IST sort of categories. So in this sort of overview, there's an IST category, and there will be sort of another slide or two that goes into a little bit more detail around this. However, there is a there's in around the ISD category, you have calcineurin inhibitors, you have anti-metabolites, you have corticosteroids, and you have mammalian target the mammalian target of a particular IST, which is referred to, which will which where the abbreviation is referred to as the MTOR. So the mammalian target of rapamycin, for example, which is sort of the brand name for SRL or sirolimus, and so one of the IST categories is the mammalian target of sirolimus or MTOR inhibitor. Now on the other side of this sort of overview, there's varying sort of brand of drugs, and these are a brand of sort of pharmacotherapeutics, and these are pharmacotherapeutics, and these are where based on the varying IST, there's sort of the brand sort of name, but also the sort of pharmacological sort of name for sort of ISTs. Now, over the sort of the years, for example, over the years, the brands may have sort of changed. However, sort of again, sort of historically or retrospectively, the brands had been evidenced as such. So, for example, CSA abbreviation for cyclosporin A, used to be under the under the pharmaceutical organization or industry, known as Novartis, and the brand name was either Nero, which was a Nero as a sort of a more sort of novel formulation, and cyclosporin A or CSA being the actual drug name, and then Sandamune used to be referred as another name for another formulation, and this sort of overview this overview around the IST sort of categories sort of lists out the different categories against the different brands of therapeutics. Now going into the interleukin 2 or IL-2 receptor antibodies and monoclonal antibodies, or MABs, as monoclonal antibodies are abbreviated. So basiliximab and daclizumab were sort of two of the first interleukin 2 receptor antibodies, or IL-2 IL-2 sort of receptor antibodies, and monoclonal antibodies. And these were therapeutics that were licensed in the United Kingdom, for example, for the use at the time of for the use in around sort of following in the run up to transplantation, but also in around sort of post transplantation as well. So these MABs or these monoclonal antibodies were traditionally administered before surgery, and they exert the IST effect only over the first few weeks post transplantation, and in recipients sort of nowadays and perhaps in some in part of sort of clinical practices, where clinical practices, although clinical practices may vary depending on where recipients might be. For example, some recipients will would have had or will as part of the workup to transplantation, or to having a transplant or an allograft, they have induction therapy, where there is induction therapy where, for example, a large sort of bolus of IST is given in the run-up to transplantation. So thus the pathophysiology, the immune system, or the immunobiology of the recipient accepts and sort of tolerates the donor allograft, once that's sort of embedded sort of surgically. So both basimiximab and daclizumab have been licensed for use in combination, have been licensed in combination with CSA and steroidal treatment, which then forms sort of baseline or mainstay therapy. And so both MABs or monoclonal antibodies work by binding specifically to part of the interleukin 2 receptor that is only expressed on activated T lymphocytes. And so inactivating interleukin 2 helps to prevent proliferation of antigen stimulated T cells. And again, sort of primitive immunology or immunobiology, primitive immunobiology or immunology is where the T cells are those cells that can cause or elicit sort of issues in recipients sort of in sort of the early phases, in the early phase, for example, in the early phase, as soon as a transplant has been grafted, grafted as or sort of again surgically. And thus, if for example if for example the the uh, uh, if for example T cells elicit a response sort of quickly, then sort of during the surgical procedure, clinicians and surgeons can see, for example, that if a sort of elicited T cell response occurs, the organ turns from its natural color into a pale or a bluish color to highlight that the organ has for example has gone into a form of a rejection, and where the color change is prominent. For example, clinicians, for example, on surgical teams would refer this as hyperacute rejection. So now a little bit around sort of in around calcineurin inhibitors. So cyclosporin A or CSA and tacrolimus or FK506 are calcineurin inhibitors that work early in T cell activation. T cell activation involves a series of cascades, and the enzyme calcineurin is one of the rate limiting points. And calcineurin is the target of both CSA and FK506 complexes, which inhibit and prevent transcription of the genes encoding for interleukin 2 or IL-2 and other cytokines that cause early T cell activation. And CSA binds to immunophilin, a cytoplasmic protein called cyclophilin A. And at the same time, FK506 tacrolimus also binds to another class of immunophilins known as FK binding proteins or FKBP, specifically FKBP12. And CSA, FK506, and sirolimus are metabolized in the liver through the cytochrome P450 pathway, and specifically the cytochrome P450 3A4 isoenzyme. And so doses of CSA used vary according to local practice or practices. And however, a typical initial sort of oral dose, oral dose in amongst the evidence has amongst the evidence in literature inform that initial dose would be around 4 milligrams per kilogram twice a day, and doses are adjusted according to pre-dose trough and whole blood concentrations. After 6 months or so, target ranges or target sort of laboratory reference ranges are reduced to aim for lower dose maintenance therapy, and and uh, and a lower dose in around sort of okay, a lower dose maintenance therapy is where all physicians or clinicians look to ensure that their patients have owing to again where the therapeutics can elicit sort of a nephrotoxic sort of effects owing to the sort of therapeutic and sort of toxicological profile of these pharmacological agents. And where this works will go into this a little bit as we go through. So now looking at sort of the IST, just some basic IST pharmacology. So you have in around IST pharmacology, or the basic sort of IST pharmacology, you have primary ISTs, and you have secondary and or tertiary ISTs, and then you have and then there are sort of there are there are other regimens or novel regimens, but also at the same time not sort of a recently not recent novel sort of regimens, but yet still have some novelty. where these agents, where these ISTs would not be utilized, or should are not utilized primary as primary ISTs, but can be used secondary or tertiary ISTs. So, for example, in the primary primary ISTs, you have tacrolimus. For example, you can have cyclosporin or CSA, and you can have mycophenolate mofetil, and then in around the secondary and tertiary IST, you can have sirolimus and then you can have corticosteroids, and then sort of an older a sort of an older sort of therapy an older sort of pharmacotherapeutic for example, azathioprine. And what would normally happen is you would have, for example, some patients or some recipients, and this some recipients, for example, owing to the differences in between for example, cross match in tissue type in around where HLA and MHC is concerned, then some patients or some recipients may be on, for example, two sort of primary immunosuppressants or ISTs, where one of the ISTs would be less would have by the by their activation through particular and specific pathways of the pharmacological sort of interplay, would be would have sort of a less potency by and as a default. So, for example, tacrolimus can be used or utilized with mycophenolate mofetil or MMF, and cyclosporin A or CSA can be used with mycophenolate mofetil MMF. But at the same time, to be transparent, tacrolimus and cyclosporin A or FK506 and neuro cannot be used and should not be used in conjunction with each other. Since these ISTs are forms of IST that are sort of the baseline or the primary sort of primary IST, and then the secondary and tertiary IST as mentioned. And what can happen with some recipients, is that they have, for example, tacrolimus, or they can have CSA or cyclosporin A, and they can have sirolimus, or SRL, and they can have recipients can have CSA, for example, with sirolimus, and for example, a corticosteroid, and can have azathioprine, which would act as basically secondary and tertiary immunosuppression. And then where there's for example, again, sort of novel regimens, but not a sort of recent pharmacotherapeutics, as monoclonal antibodies, as per mentioned, basiliximab and daclizumab, and sort of pharmacotherapeutics SDZ RAD or RAD, which is a derivative of sirolimus, or SRL. And there's also FTY720, which is an antibody preparation, and there's beta letacept, which is a derivative of a CTLA-4 IG, which is part of a pathway in the way biletacept sort of functions. And so IST, patients or recipients can be on sort of mainstay therapy, and then can be on dual therapy, where clinicians refer to a second sort of where recipients are on primary or dual therapy, and then can be on tertiary therapy. So where a patient or recipient can be on two for example, two or three immunosuppressions, IST plus two or three IST plus a corticosteroid. A corticosteroid, by the way, is not sort of under the basis of a of an IS under the basis of IST, but corticosteroids do have a sort of an inflammatory sort of effect. And so some pediatrics can be on dual or triple or even quadruple treatments, for example, sort of the differences or the differentiations between tissue typing and cross matching in around where transplantation and allografts are concerned, but also owing to for example the etiology sort of as part of initial diagnosis of where organ failure has been or is a basis. So now going into a little bit around biomarkers. So biomarkers are very are kind of are sort of an intrigue generally, because there's been sort of in the last in the last decade or so, there's been a lots of sort of works published by varying authors, looking at varying sort of biomarkers to look at sort of renal function, but also how different biomarkers have a basis in also sort of helping or supporting sort of laboratory practices or laboratory specialists to ensure that the appropriate IST and trough levels are matched, so that patients do not, or recipients who have been allografted, for example, do not get, for example, those sort of nasty nephrotoxic those sort of nasty nephrotoxic sort of side effects from these already potent pharmacotherapeutics. So biomarkers of kidney damage are affected by several factors, including age, but certain populations, for example, children or young people and older adults, may require special consideration, as the normal laboratory reference ranges can be or are elevated. And so in pediatric recipients, serum creatinine or SCR reach sort of adult levels by the time they hit adolescence. And so moreover, these recipients are at higher risk of developing drug-induced nephrotoxicity, and simply because partly to do with again how the trough levels can be challenging for both sort of clinicians and physicians to get right for a pediatric recipients, but also where, for example, the pharmacotherapeutic sort of profile of many of the ISTs IST or ISTs have sort of underlying underlying effects to an extent that complicate or can complicate the way sort of pharmacotherapeutics are dosed initially, and so the different biomarkers for renal function, but also for monitoring sort of mainstay IST include, for example, cystatin C, number one, cystatin C, two, estimated glomerular filtration rate, three, glomerular filtration rate, four, number four being one of the more novel, one of the sort of more novel biomarkers to an extent, neutrophil gelatinase associated lipocalin, and number five being SCR, again, serum creatinine. And so cystatin C or cystatin 3, formerly gamma trace post gamma globulin or a neuroendocrine basic polypeptide, is a protein encoded by the CST3 gene and is mainly utilized by a biomarker, that has been utilized as a biomarker for renal insufficiency or renal function. Serum titers of cystatin C are a more precise test for renal, a precise test and around renal function, as represented by EGFR and GFR than SCR, serum creatinine. And cystatin C titers are also less dependent on age, gender, ethnicity, and other sort of parameters compared to creatinine. So now, now looking into sort of comprehending drug-induced nephrotoxicity. So early investigations surrounding transplantation had led to attempts to understanding of renal immunobiology. Unfortunately, at the same time, what also had been realized was that that no effective sort of IST agents at the time have sort of achieved a desired effect, where, for example, nephrotoxicity does not where nephrotoxicity sort of does not occur. So the first attempts of IST involved for example, total body irradiation, and although radiation prevented transplant rejection, this caused aplasia, which was responsible for a high mortality rate. And then anti-neoplastic drugs or anti-neoplastic therapeutics, such as, for example, methotrexate and cyclophosphamide, were used or utilized for IST, but these agents proved to be sort of toxic and were associated with poor outcomes. So in sort of more retrospectively, in the 1960s, prednisolone as a corticosteroid was utilized to control a severe episode of rejection, and subsequently 6-mercaptopurine and its sort of successor, at least in part, azathioprine being in line to sort of hierarchy. Azathioprine has sort of a hierarchy over prednisolone, and was tested successfully in transplantation investigation involving canines, which where investigations were carried out by by an early investigator, Roy Kahn, and were introduced into clinical practice. Eventually, azathioprine and steroids were utilized as a combination therapy. And despite some initial successes, the rate of early graft or allograft failure was high in renal transplantation, and only a handful of kidneys function longer than 1 year, longer than for 1 year post transplant. So most of the successful cases involved allografts, again from live related donors, since from live related donors. Hence why in literature amongst the evidence there's been a lot of prompt around live related donors. And again, as per mentioned in early parts of this interlude, the in the sort of mid 1980s to the mid 90s, the allograft prognoses between live related donors and cadaveric related donors were known to be and evidenced as comparable in amongst sort of investigative works. Now, at the same time, in around sort of a critique of laboratory medicine and around around these sort of biomarkers, the renal biomarkers, the renal biomarkers, nucleated gelatinase associated lipocaine and cystatin C have emerged as potentially useful or helpful indicators for GFR of GFR, with sort of the latter postulated as sort of a gold standard. Now, at the same time, before going on, creatinine or serum creatinine have been biomarkers that have been utilized to in around renal function or around renal function or renal insufficiency for many years. However, cystatin C, in investigations and published works by authors have indicated that it has some superiorities that creatinine has sort of where creatinine or serum creatinine sort of negates. And so at the same time, sort of nucleated gelatinase associated lipocaine is a 25 kilodalton protein bound to gelatinase originally identified from neutrophils, which is also expressed at very low levels in several titers, including the kidneys, and is released systematically in response to renal epithelial damage produced locally in and through renal tubules. So, nucleated gelatinase associated lipocaine also undergoes glomerular filtration followed by tubular uptake, and is one of the earliest and most and sort of one of the sort of stronger sort of induced proteins in the kidneys after ischemic or nephrotoxic injury in animals. At the same time, nucleated gelatinase associated lipocaine can be easily detected in blood and urine post transplant, post transplant, and hence in parts of the world and parts where authors and researchers have published works have informed that have informed that 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 nucleated gelatinase associated lipocaine is an emerging is and has been emerging as a major predictive sort of biomarker with potential use in risk stratification of EGFR, of EGFR, CKD progression, looking at CKD or chronic kidney disease progression, and immunosuppression therapy or IST monitoring, and in around transplant rejection. So now a little bit around sort of getting the toxicology profile right. So the pharmacokinetics of IST such as CSA or cyclosporin, FK506, MMF, and sirolimus are complex or multifaceted and unpredictable. It is also important to identify a specific pharmacokinetic parameter, a pharmacokinetic parameter, at least where laboratory medicine is concerned for each individual pharmacotherapy, whether it is trough levels or area under the concentration time curve, which may be the most useful as a basis for optimal TDM or therapeutic drug monitoring in clinical practice. So now, sort of an overview, in around as a summary. So the direct nephrotoxins. So for example, gentamicin being from the aminoglycoside sort of family of drugs, and historically have been utilized to treat pediatrics or young people for severe who have severe, for example, UTIs. Non-steroidal and sort of therapeutics, anti-inflammatory sort of pharmacotherapy, rifampicin, paracetamol or acetaminophen, contrast media, for example, including sort of radiopaque substances, carbon tetrachloride, and ethyl glycol, and different IST efficacy and safety profile can have a toxicological impact in pediatric renal recipients, also depending upon the disease etiologies and pathology. And so it remains a challenge. To establish peak IST therapeutic dosing. Novel IST or new IST are designed with the objective of lowering regimens and doses, which should also reduce nephrotoxicity whilst maintaining sort of therapeutic potency. However, one of the main points here is how more crises exists regarding immunohaematology and immunogenetics, and where serology surrounding transplantation for pediatric recipients have been launched into as a factor with IST to prompt prognoses. So at the same time, TDM should ensure that concentrations are not too high or too low, thereby reducing the risk of toxicity and the risk of toxicity and precluding AAR, acute allograft rejection. And also in literature there have been investigators looking at donor specific antibodies or DSAs, and DSAs remain an issue amongst pediatric recipients. So deeper understanding of the pathophysiological mechanisms and development of novel compounds regulating sort of regulating or in sort of synchronicity with the immunobiological basis of recipients and the response and the sort of the humoral response are of intrigue to improve transplant outcomes. And that ends this audio podcast. So thank you for tuning in on this interlude on this interlude as part of a Storm Robotics production.